Using bacteria to make copies. First of all, you need to isolate your DNA. Production of DNA fragments that have the required gene using reverse transcriptase or restriction endonucleases. So for example, if you have a sample of cells with the DNA, you can use restriction endonucleases to chop out the piece of DNA that you wish for. Alternatively, use a cell that you know produces that protein, because it will contain lots and lots of mRNA for that protein. Extract the mRNA and using reverse transcriptase, make many copies of that mRNA into cDNA. Then, using DNA polymerase, make double-stranded DNA. An alternative method to these two is to synthesize your own gene product using a gene machine. In the gene machine, you would have a base sequence that you have determined because you have a polypeptide. So you identify the amino acids in that polypeptide, then, knowing the most common triplet sequences of DNA bases used to code for each of those amino acids, you make up the base sequence. You then place that into the gene machine and it makes oligonucleotides, which are small fragments of nucleotide strands based on the base sequences that you have typed into the gene machine. The gene machine then uses algorithms, which are mathematical equations, to work out which fragments of the oligonucleotides need to overlap and be joined together, and in so doing is able to make a long sequence of DNA that can be used and inserted to modify an organism's using vectors. So, once you have your piece of DNA, you then need to insert it, and you can insert it into a vector. An example would be a plasmid. You would cut the plasmid using the restriction endonuclease, and using the same restriction endonuclease, cut the piece of DNA, adding with it promoters and terminators and sticky ends. Next, transformation. Introduce the DNA fragment into the suitable host cell. Once you have ligated your plasmid with your piece of DNA, you would have a recombinant plasmid. That recombinant plasmid needs to be taken up by the bacterium. That is done by introducing calcium ions to the bacterium and changing the temperature. Some bacterium will then take up the recombinant plasmid, however some will not. And that leads us to our next step, identification. Identification of host cells that have taken up the DNA using gene markers. In the case of a plasmid, you may find that the plasmid contains antibiotic resistance genes. If you have used a restriction endonuclease enzyme, it will cut in the region of your antibiotic resistance gene, therefore rendering that particular gene useless. You then insert your piece of DNA that you are interested in. The plasmid closes back up, but it also contains a marker gene, a gene for antibiotic resistance, that is still intact. Using those two genes, the broken antibiotic resistance gene and the intact antibiotic resistance gene, it is possible to identify bacteria that have no plasmid, as they will die on any of the antibiotic plates, a bacterium that has a non-recombinant plasmid, because that will not die on any of the plates, no matter which antibiotic you use, because they still have both genes intact, and then finally the recombinant plasmid in the bacterium. Those bacterium will be able to survive one of the antibiotics, but not the other, and by differentiating between the two, it is possible to identify the bacteria that contains your recombinant plasmid. This is identification, and the antibiotic resistance gene in this case is called a marker gene. Other marker genes that you can use include fluorescent markers and enzyme markers. And finally, growth and cloning. Once you have identified the bacterium using gene markers, you must then culture the host cells containing the DNA to produce the protein on a large scale. In the case of the insulin protein, you would isolate the insulin and it can then be used in humans to help them with their everyday lives where they do not make their own insulin. Fluorescent markers are genes that code for the green fluorescent protein. That can be inserted into plasmids, and if they take up this particular gene, they will fluoresce green under UV light. It is relatively simple and straightforward to identify the bacteria by simply passing over a UV light over the top of them and they will glow. Other alternative markers include enzyme markers, genes that code for enzymes. By adding a substrate to a petri dish, it is possible to identify those bacteria that contain the genetic marker gene for an enzyme, because if the substrate is a blue colour and then it turns colourless, or vice versa, it is possible to identify those bacteria that have the enzyme because they will be able to cause the colour change in the substrate, whereas bacteria without that enzyme will not be able to cause a colour change, and so therefore identify the bacteria that have been modified using the recombinant DNA.